All right, here we're continuing with the PowerPoints for Chapter 5. You must know what isotonic, hypertonic, and hypotonic are. I will provide a video on osmosis, perhaps mine. I have one, but actually there are probably some really good ones out there. I'll look around and see if I can find some with more animations that help with understanding isotonic, hypertonic, and hypotonic. So make sure you know these. Understand that essentially what you're looking at here is how solutes get in and out of the cell. The idea is that depending on the situation, osmosis may allow certain things in, or it may force things out of the cell. So I have a story called the catfish story, which I will make a video on, and you can learn it that way. I'll also try to find some more videos that have to do that have more animations in them than I can provide. All right, read through this. Of course, things can come in and out, mainly water. That's what we're talking about here. And depending on what else is in the cell, some things draw in water, other things draw out water. And then there's the case where it's in equilibrium. The term equilibrium, would be isotonic, where water or whatever is moving is not moving really. It actually is, but it's equaled and it's not causing the cells to swell or to shrink. So keep those terms in mind when you hear me tell you something like that, like this term isotonic. You should right here, you should be aware of, well, can't highlight it. You should be aware of this term. All right, now read those. Read this. Know what lysis is. Essentially, a cell explodes if it lyses. Plant cells. It's really important for plants. Realize that a plant cannot get up and move around if it runs out of water. It has to grow its roots or whatever, or it has to conserve. So plants care a lot more about water than animals. Animals tend to be able to be mobile and can go find water. Plants, they rely on water heavily, and they must learn how to deal with it when it's not there. You can skip the part on the protists, so we're not interested here. But transport proteins are very important. Some things can move, like water moves via osmosis and diffusion, but there are these pumps that we've talked about in previous videos, and they can be classified as either channels or transporters. So channels you don't think of as being powered usually. Transporters you do, but really the terms that you want to use, if ATP is used, it's going to be what we call active transport. All right, and that means you used ATP or energy. If you didn't have to use ATP, that is passive transport. You shouldn't necessarily call it passive, but it's a different type than active. It's not active. All right, so here we go. We're looking at these proteins here, and as you can see here, it's closed. There are two proteins, one here and one here, and they go all the way through the membrane. And when it's closed, it looks like this. Now, certain things can cause these proteins to change shape, and when they do, it opens up a channel. All right, so it is gated if it can open and close. All right, now often there are binding non-covalent, that means electrostatic, hydrogen bonds, that sort of type of thing, can bind to these proteins. And when they bind, they can alter the shape of the proteins or make them move. And when they do, then the channel opens and closes. And these small molecules that stick to the proteins are called ligands. You need to read this. An aquaporin is exactly what it is. It's a pore that lets water move through it. You need to know the term lysed. You need to know what hypotonic and hypertonic and isotonic mean. All right, so you need to know what an integral membrane protein is. So, as general, you need to know those terms, and you need to understand osmosis. All right, and then you need to understand transport proteins and facilitated diffusion. What that means is you facilitate the movement of water across the membrane. So these are just some terms. Get used to using those terms. You do not have to actually read this in detail. I won't ask a detailed question on that. But you do need to know the terms that come. If you do understand it, then you will have learned the terms. So there's a reason to maybe go and read it. Understand conformational change. In other words, conformation is kind of like the changing of shape of the structures. In other words, if you want to bring in certain types of molecules, often you're going to have to use some energy, or at the very least, your proteins are going to have to change their shape in some manner to get that material into or out of the cell. And that's what they're talking about here. And these proteins, or these pumps and pores, are often called transporters. So just realize what a transporter is. It usually gets something across the membrane. And there is a uniporter, so that is a single molecule or ion. All right, so it just moves one thing at a time. There is a symporter or contraporter. In other words, it only works when you've got two of something or more. All right, and it often will let one thing in if you let something else out. A little bit more complicated. Or if you've got two things together, then it lets them in. This example is a symporter. So in other words, two things need to be present for them to both get across. In other words, they both can transport across, but they both have to be present before it's going to work. That is one type of transporter. That is a symporter. Contraporter would be when you have one has to come in and another one has to, or actually, yeah, that's an antiporter, sorry, where something has to come in to let something else out. And they do not necessarily have to be on the same complex. So you can see here that the little blue molecule attaches here, while there's another one over here that's red. So this let this one in, but you had to let one of these out in order to get this to continue. So there is an antiporter, a symporter. Contraporter could be like both of these in the same one, but we won't worry about it. Just symporter and antiporter will work. And then there's a uniporter. So make sure you know those terms for the exam. Active transport, of course, means it needs energy, and for the vast majority of the time means ATP. So ATP must be present, and you must understand that concept of ATP being converted into ADP, all right, adenosine triphosphate turned into adenosine diphosphate. In other words, it loses one of the five phosphates. Tri meaning 3, and di meaning 2, so adenosine triphosphate versus adenosine diphosphate. And when it gives up that P, that phosphate, off of one of the adenosines, releases energy, and that energy goes to the protein, and that protein can then change shape and do some work. You must understand that concept. There will be lots of questions about that. Primary active transport and secondary active transport. So this uses a pump directly and uses energy to transport things. So it's a powered pump. Just as we've seen things that are transporting things, you can consider those pumps. And active transport, primary, it just means we powered it with ATP usually. Secondary active transport uses a different gradient using a pre-existing gradient to drive transport. Well, gradients are like there's a whole bunch of one thing on one side and a bunch of something else on the other side. And it causes they would like to mix, but you've intentionally separated them. And so there is a desire for these to move to the other side. It's a natural chemical reaction. And you can use that if you put certain things into the membrane that will let that happen. And so this is a secondary active transport where you use a gradient, or that you pump against a gradient. So you need to understand this term gradient to understand the secondary active transport. All right, so here is a pump that actively exports protons, H+. So you need to know that that is a proton. A hydrogen with a plus sign on it is a proton. And here you have, this is outside the cell, this is inside the cell. And so and notice we are using ATP here. And what you are doing is you are, pumping hydrogen outside the cell. But then when sucrose is available, you can use the hydrogen gradient. In other words, you built up a bunch of hydrogen in here and it all wants to get out. And you can use that to pump sucrose against the gradient. I'll let out 2 hydrogens if you'll let in 1 sugar, is essentially what we're saying. So the hydrogen has built up in here and it wants to get out. And chemistry will allow you to say, let a sucrose in if you let 2 hydrogens out, or something like that. A certain amount of hydrogen can get out. We'll let a basically something else to get in. All right. Don't worry about this one. Of course, it's messed up here on my thing, but there are ion pumps, and that's hydrogen is an ion, and H+ is an ion. It's a proton. Or you could be doing electrons. But the whole idea here is keep in mind what's going on, and the ATP is involved. I'm not going to ask you a question on this in any detail. You don't have to worry about that one. Now, sodium ions. Now, this is not just as simple as a proton. H+ is a proton. Na+ is a sodium, so it's a much more complex molecule. It's not as simple as a proton. It's much larger, a lot more properties that are needed to do something like sodium pump. So sodium pump is going to be much more complicated than, say, just a hydrogen pump. Take a look at this. I may ask a question. Probably something having to do with how you get sodium in and out. This is very important when you get to the nervous system. Notice this is in a nerve cell. And so if you take additional classes like anatomy and physiology, you will need to be familiar with this. So if you're planning on taking something, if you're in nursing or something like that, this is an important one to learn. So I do ask questions on this one. So make sure you look at this particular drawing and be prepared to answer some questions. Now realize you're not just moving sodium here, you're also moving potassium, K. And this is, again, you let 2 in of one type, you let 1 out of another, plus they're acting as ligands. All right, so there are a couple of things here. I might do another video just on this. If I decide to make this a major essay question, I'll make a whole new video on it. It may just be multiple choice questions, though. All right, you absolutely must know how to do endo and exocytosis. There will absolutely be several questions involving endo and exocytosis. Now, how are these different from pumps and pores? Well, this is when you package some of these proteins or ions, whatever, it could be water. You package them together, all right, into like a lipid container, a vacuole, or whatever you want to call it. You wouldn't call it a vacuole, you would call it it's called a vesicle. And it's like a little ball, and inside the little membrane ball you package something, and you want to expel that vesicle outside the cell. So you can package up things like hormones or maybe wastes, and you basically can dump them out into the matrix. And so this is how we do that, and we use something called exocytosis when we want to get rid of things, all right, or at least export them outside the cell. We want to bring them in, we do endocytosis. And so I have some videos that I've made, plus I can get some links to some more animated ones for you to learn the difference between exo and endocytosis. These will be, along with pumps and pores, major questions on the exam. All right, now here's what we're viewing. So here is the cytosol. So we're inside the cell right here. And so here we package our waste product, whatever it is, these little triangles, these little yellow triangles, we package them inside a little ball of membrane. And they do put a kind of a protein coat around it to I guess, protect it or signal that it needs to get outside of the cell. And then it merges with the membrane, all right? And then it just kind of pops out when it's made right, all right? In other words, this thing could pop out. That would be exocytosis. Now, endocytosis is over here. So what happens is this molecule kind of sticks. Maybe there are some proteins here that they're not showing, but this molecule will stick to the membrane, and when it does, it causes conformational changes in those proteins, and that changes the shape of the membrane, and what's called an invagination occurs, and basically it kind of makes a dent, if you want to call it. It starts out that way in the membrane, but eventually that dent kind of folds in on itself and actually forms a closed circular structure that can then bud off from the membrane, and then it loses this protein coat. A lot of these proteins are the things that they stuck to, and they are linked, and so they'll break apart, and then you can do something else with it. You can get whatever this is, you can now bring it into the cell in large quantities. So the whole idea is endo and exocytosis. Move things in and out of the cell in large quantities, and that's what you want to remember. That will be the kind of question I ask you, is what type of process would you use to move large quantities of something, or a large individual thing in and out of the cell. And you would, the thing that can move the most and the largest things that are the largest structures, so like proteins, hormones, those sort of things, those will typically be moved through endo and exocytosis. Individual proteins perhaps can be done by pumps and pores, but generally pumps and pores usually move molecules, individual molecules that are not very large. And then with osmosis and diffusion, that's usually small things like water and some ions, maybe some solvents might be used there, or block that. It's related. So as you get larger, the smallest things, mainly water, are going to be usually moved by osmosis. But you can have things smaller than water that are pumped. Certainly ions can be pumped across active and passive transport with the pumps and pores and channels, things like that. And then, but when you're talking about large quantities of something or a larger molecule, you're probably going to end up having to do it using something like endo and exocytosis, which is what we have right here. All right, and you need to know these terms. Read about receptor-mediated endocytosis, pinocytosis, and phagocytosis. So you'll need to know what all three of those types of endocytosis are. And realize these can be reversed, and instead of it can be, instead of endo, there could be exo versions of these. So here we go. This is kind of the reverse here. The receptor. So you want to get this, this is what I was talking about in the last one. You might have these proteins that are in here that these can stick to. So you want to get this inside so it sticks to the protein receptors here. And that causes a conformational change so that it forms this, and then it pinches off and then comes into the cell. This one is more or less showing you it getting out. this one is showing you coming in. So keep in mind what these two are. These two figures, they will be something like them will be on the test. In the past, I'll give you an idea. I have given this picture without any writing on it. In other words, this might say cytosol, but the 1, 2, 3, 4, 5 are all gone, and I will ask you to explain what is going on in this picture. So if you know these 1, 2, 3, 4, 5, you'll be able to answer that essay question. Similarly, I could do it with the other one and take off these and ask you, just give you the picture, and then you explain to me what's going on in that picture. That's what I have done in the past, just to give you an idea of what is possible. So this is it for this chapter. Mainly it's this second half of the chapter is about getting things in and out of the cell. The first half was more of like the structure of the cell. So it's part 2 of this chapter. I did one by giving you the overview. So there's part 1, but then there's part 2 and 3. And 2 and 3 break up the actual chapter here in PowerPoints. And really, to me, this should be 2 different chapters. One about the structure of the membrane and the other about transport. But they decide to put it all in one chapter. All right, that'll do it for this one. There are a few more things I want to talk about for this exam, but we are getting close to having another exam.